Good morning, everyone. This is Kathleen Arobo,
And I, Hannah Princess Arais,
and now we're going to discuss our topic, which is the mutations.
and it's all about mutagenic agents.
The factors which can cause genetic mutations are called mutagenic agents. Mutagens have a different types, and the first is physical mutagen, chemical mutagen, and biological mutagens. Mueller 1927 was the first to find out physical mutagen in Drosophila.
Mutagenic just refers for an environmental agent that damages in DNA, so in there increases the rate of mutations. In this video, we're going to chatting about the different kinds of mutants, which is links up with the syllable stop points. Shown below the video, there are lots different mutagens in our environmental, but there are three main groups, which is we need to look at electromagnetic radiation sources, chemicals, and naturally occurring mutants. So guys, you know that the drill, let's get it into. So firstly, electromagnetic radiations. Radiation refers to a group of waves which carry electromagnetic energy. These waves include radio waves, microwaves, infrared light, visible light, UV light, X-rays, and the gamma rays. Now, don't get me wrong, of all these guys are a part of our radiation club, but not all of them are a part of mutagen club only. These waves at the side of spectrum, pseudo ones, which are short wavelength, high energy waves, can cause mutations, so they are the only ones we really need to care about. Okay, so we need X-rays, gamma rays, and all the type of radiation which cause mutation. But now how they do actually cause mutation? Well, the answer comes down in electromagnetic waves are carrying a lot of energy when they are though metal is like a cell, or example, they give up some their energy, so atoms heat the atoms are now super hybrid up, charged with the extra energy from waves then literally vibrate with excitement, just like someone who's had coffee and cheese is with a couple of red balls on the top of vibrating. In a time of loss of some energy, and atoms will also lose electrons. This is what the causes of bonds to break the causes damage. So for example of chemical bonds, which holding then it backs together might break as to the backbone's plates, or the bases might be replied into wrong. Some cases the cell DNA will be so damaged that is the dies, but it is also possible that the cell may survive, only that some of the DNA is mutated. So overall, you are really want to avoid high energy electromagnetic radiation, the daily life, but the thing is, unless you're living under the rock, you can't avoid. The biggest source of electromagnetic radiation in the everyday life is the sun. It is emits infrared, visible, and UV light that make it to the Earth's surface. Even though infrared and visible light aren't strong enough to cause mutations, the dangerous one, which I'm sure you're all well aware of, is UV light, which can cause mutation. This is why your parents have been nagging you to wear a hat and sunscreen your whole day life. Basically, the sun is dangerous. Best who really don't wanna miss with. Okay, so another source of high energy radiation, last common source of electromagnetic radiation, with mentioning a medical imaging machine like X-ray machines and pH scanner. X-ray machines are also used as airport scanners. The second big group of mutants are certain chemicals. Different chemicals can cause mutation, but they do it in every different ways. So instead of doing a big explanation, we're just going to look at an example which cover the main ways chemical causes. There are a number of chemical mutagens. Some are known as bases analog. They are similar to DNA bases, can be incorporated into replicating molecules, but deeper with incorrect nucleotides. So for example, 5-bromouracil will replace thymine and can be bind to guanine. Some chemical mutagens are known as hydroxyl iding agents because they add no H group, a hydroxyl group. If a hydroxyl group was added to cytosine, this would be then caused to bind. Some chemical mutagens are known as alkylating agents. They can add either an ethyl group or a methyl group, and this will change base pairing. So for example, if an ethyl group is added to guanine, it will then bind to thymine. Some chemical mutagens are known as demyelinating agents because they remove animo groups. If an animo group is removed by cytosine, it's converted to a uracil and will bind to adenine. If an animo group is removed from adenine, it will become hypocytine and will then bind to cytosine. Some chemical mutagens are known as intercalating agents. After interacting with the DNA, they can cause base pairs to be deleted and they can be caused base pairs to be inserted. Now, this is a particularly dangerous in coding regions because insertion and deletion are frame-shift mutation, which can then alter all of the downstream codons.
Okay, so the next is the cell regeneration. It is the natural process of replacing or restoring damaged or missing cells, tissues, organs, and even entire parts to full function in plants and animals. This is some example of cell regeneration. Hybridization. At high temperature, double-stranded DNA will denature or separate into single strands. When the temperature is lower, the two strands will annihilate because of the base pairing interactions of the complementary strands. If the nucleotide sequences are similar, complementary strands from different sources will also annihilate. This is called hybridization. Indicate that each strand of DNA came from a different source regions of different stranded DNA in which the respective single strands hybridized to one another are said to be homologous or have similar or identical nucleotide sequence. The high specificity of base pairing interactions between complementary strands of DNA can be used to locate a specific nucleotide sequence in a sample. If the DNA from one source is immobilized by attachment to a solid surface, such as nucleocellulose homologous DNA from another source will hybridize and be retained by the immobilized DNA. This is the basis for various DNA probe techniques. Non-homologous DNA will not attach, and that is hybridization.
So once again, good morning. Since we tackle our topic that is all about mutagenic agents and excluding its types,
Ionizing radiation, chemical agent, cell regeneration, and hybridization.
then happy watching!